Assalamu alaikum. I am Dr. Jamil Abbas from the Physiology Department of the Faisalabad Medical University. Dear students, how are you? In continuation to my previous lecture on the countercurrent mechanism, and we have discussed the role of sodium and how the sodium is get accumulated in this area, that is this inner medulla, how this hyperosmolarity is produced for the first time. We have discussed this one, and we have discussed that what are the peculiarities, what are the very important particular things concerned with the descending limb of the Lupa family, and we have seen the very important properties of the ascending limb of Lupa family, particularly this thick segment of the ascending limb of Lupa family. And we concluded that this hyperosmolarity of this area, that is the hyperosmolarity of the medulla, is due to the accumulation of the sodium, and the main role was of the your sodium potassium 2 chloride channel protein present over here. But remaining 40%, that 60%, on some books say there is 50-50, but some books say 60-40. Roughly we say that 60% of the accumulation, or you can say the production of hyperosmolarity of the medulla, is because of the accumulation or deposition of the sodium into this area. And this sodium comes from this area through sodium potassium 2 chloride channel protein, and some of this sodium do leak out from this area, the passive diffusion from this area. Now let's discuss the remaining 40%, from where that remaining 40% of the hyperosmolarity of this area, from where it do come out. Actually, now the role of urea we are going to discuss. Now urea, the name urea, it is basically formed in the liver. Although it is a topic for the biochemistry, that how it is going to form. But uremia, literary meaning of this uremia, that means urine in the blood, but actually we don't take it in this meanings. We take the uremia, that means high urea level. The normal urea level in our serum, in our blood, is 40 milliequivalent per deciliter. If this urea level is more than this, then we will label it as uremia, and this is the term in the usually occurs in renal failure, because production of this urea is done in the liver, and this urea is coming from the protein diet. I'm not going into details how urea is going to produce, but this urea basically produced from the ammonia, or further you can say it's coming from the ammonium ion NH4. Ammonia or the ammonium ion, they are highly dangerous or more toxic as compared to the urea. So in the liver, your more toxic substance, that is ammonium or the ammonia, they are converted into a less toxic substance, the urea. And once the urea is formed in the liver, now it is the prime responsibility of our kidneys to excrete it from the body, to remove it from the body. And 80% of the urea produced by the liver is removed from the body through these nephrons, or you can say the kidneys, while the remaining 20% is removed through your GIT. Now let's see how it's going to be occur. Now urea, once it enters into the filtrate, now here you see the 100% of the urea, that means this is actually 4.5 milliequivalent is the concentration in the filtrate. This remains 100% in this area. This remains 100%. A very, very small amount do reabsorb from here into the blood, into the peritubular capillary network, mainly through the solvent drag mechanism. You know that there is a transcellular mechanism, there is a paracellular mechanism, and there is a solvent drag mechanism. I have told you in your classroom lectures that what is meant by the solvent drag. Just like the wave of water, by the wave of the ocean drags the sand or the other thing in it drags from the bank of the river or bank of the ocean into the mainstream. Similarly, solvent drag, the water is going to be reabsorbed from here between the cells, and when the water is moving between the cells, it also drags some of the urea into the peritubular capillary network. So some of the urea do moves, or you can say do reabsorb in the peritubular capillary network in the proximal part of the proximal convoluted tubule of the nephron. But you will see the arrow is showing in the last part of the last part of the PCT. Here is the last part of the PCT, not from here, because here the reabsorption first occurs the sodium, then the prime responsibility of the sodium to take the glucose along with it, then the second responsibility is the amino acids, and after that the chlorides and the water reabsorb itself. While all this thing is happening over here, the concentration of the urea is going to be high because water is leaving out. And when this concentration becomes high in this portion, some of the urea reabsorbed through solvent drag mechanism. And the remaining urea, it enters into your descending limb of the loop of Henle. This is your diagram from your guidance. It's moving down, and here you have seen the previous lecture, the water, this area is highly permeable to the water, and the water is going out, is water is going out here, it's going to be deposited over here, sorry, reabsorbed in this area. And while the water is reabsorbed, here inside the concentration of urea become high and high, become more because water is coming out. Now as we take the U-turn, and I will came back to these arrows. First I will moving up into the thick ascending limb of home loop of Hamlet. Here is a dark orange, or you can say dark brown line. It is you can follow this line according to the cursor. This area is impermeable to the water, but this area will become permeable to the water under the influence of ADH, anti-diuretic hormone. Now this area, and by the time filtrate reaches over here, now the urea is not going to be reabsorbed from this area. You will see there is no urea is moving out. Here the urea 100%, and due to the water 50% was reabsorbed in this area, remaining 50% due to the movement of the water out of this area, it again becoming 100%. And why the things are adding over here, let's see later on. Now the urea, it's moving up through the ascending limb of the loop of Henle, and this tube is not permeable to the water. It will become permeable under the effect of ADH. And this part, distal convoluted tubule, it has 90% cells which are known the principal cells. These principal cells are the site of action of the aldosterone. These principal cells, I am not changing my diagram over here, but just have an idea that this area have got the 90% principal cells, and 10% we have got the intercalated cells present in this area. And these principal cells, they are responsible for the reabsorption of the sum of the sodium from here under the effect of the aldosterone. Basically, aldosterone is the basic function of the aldosterone is to excrete potassium, although it's going to reabsorb the sodium. But the aldosterone also causes the main function of the aldosterone to excrete the potassium ions from the body. Let's see later on all these things. Now the urea is traveling. Now it become again 100%. How it become 100%? Let's see later on. Now we are dipping down again from the outer medulla. We are moving down into the inner medulla. Here this portion is permeable. Actually, this is not the permeable. Actually, we have got some channel proteins present over here, which are known as urea transporters. Actually, basically, there are three types of the urea transporter. Urea transporter A1, A2, and A3. Here we have got urea transporter present over here. Through these urea transporters, the urea moves out in this way, and it travels into the inner medulla, and this area is again permeable to the urea and re-enters into this loop of Henle. So this addition of the urea, because this area is permeable to the urea. So urea is coming out through the channel proteins, urea transporters which are present here, and here we have got actually the urea transporter A3 which are present over here. They are responsible for the movement of the movement of the urea inside this area. So once it re-enters over here, and again it's going to move up. Now under the effect of ADH. Actually ADH has got its, we will see later on in detail the effect of the actions of the ADH. Basically ADH is going to form a channel protein which are known as aquaporins. And at this stage you can understand that this area become permeable for the reabsorption of the water under the effect of ADH. ADH causes the pure water reabsorption. There is no sodium involved, no sodium reabsorption under the effect of ADH. So when the ADH comes, antidiuretic hormone, it causes the absorption of water from here to this side, from here back to the peritubular capillary, from here back to the capillary tubular network. So the water is going out and urea become more concentrated. As we move over here, here we have got the aldosterone action on the principal cells in these areas. Aldosterone causes the reabsorption of the sodium. So the urea become more and more concentrated. And by the time it moves down, it moves down. Now it's highly concentrated filtrate, or you can say high concentration of the urea present over here. High concentration means more urea, less water. More urea, less water. So urea again leaks due to high concentration over here and less concentration over here. It comes out through their urea transporters A2 on A1. They come out, they do come out, and along with it, the action of the ADH on, although we have seen the action on this area, the pure water reabsorption under the effect of ADH. But this ADH also causes the movement of this urea out of this area into this area. So the ADH has got basically two functions. Although ADH got a lot of other functions, we will see later on. It's called the vasoconstriction. That's why the name given to it is the vasopressin. But I am not going to detail the ADH at this time. But at this time, you must understand the ADH performs two functions over here. Although ADH has got a lot of other functions, but concentrating on the two functions, that the ADH causes the reabsorption of water in these dark areas, dark brown line, and this will assist in the high concentration. That means urea become more and more over here. By the time it takes a turn, it become more urea, less water. But here, the reabsorption of the urea along with the water, this urea outward movements also stimulated by the ADH. And this movement of the solute or main solute, that main solute is your urea and the water, all of it. So remaining, some of it's going to be, you can say the 80% is going to be moved into the interstitium, and 20% remaining is going to move out from the body. This is excreted in the urine. But before excretion, it will take a turn, it will take a lot of cycle, it will move again and again, again and again in this way. It's going to move in this again and again. And in every circle, urea become more concentrated. Urea become more concentrated in this area. Although it's highly concentrated, but the movement of urea out of it along with the water, it doesn't mean that the more water is moved out and less urea. No. Urea and the water, they move out in the same ratio. That's why concentrated urea 80% moves in this direction and 20% is excreted in the urine. But by the time it is moving in this direction, it makes this area, the inner medulla, more and more hyperosmolar. So this movement, this outward movement of urea from this side and traveling of the urea through inner medulla into this side contributes in the formation of the hyperosmolarity of the medulla. And medulla, this urea recycling, urea is in this way, it's moving in this way. This urea is moving in this way. Actually, this urea recycling word is used by the biochemistry. There is something different. Here, the recycling is, you can say, again and again recircling of this urea. And you can well understand if I make a diagram, or you can say, if suppose if you are driving a car and moving, if you're driving a car in this way, from follow my cursor, and you are moving here, and here you have got a roundabout. Here you have got a roundabout, and after that you move onward. Now, if government orders you to take 4 or 5 rounds of this roundabout, then you will be allowed to leave this area. Then what will happen? Then you will see a lot of traffic over here, because you have to take more and more circles around this roundabout. It is ordered by the your government. That's why, once, if it is this recycling or this movement again and again is not there, then this concentrated urea with less amount of water will not be possible by the kidney to excrete it from the body. Now you can well understand that how urea contributes, that is 40% of the hyperosmolarity of the medulla is coming by the from the movement of the urea from this side to this side. Now I'm going to read all these things. A normal person can excrete about 40 to 60% of filtered load of urea. It contributes 40% or the 500 milliosmol in the hyperosmolarity of the medulla. Urea excretion is equal to the urea level of the plasma multiplied by the GFR. Now this, you can say, equation is very important. It's coming from the guide. Very important. That if the kidneys, they are damaged and their GFR is going to decrease, their GFR is going to decrease, is less GFR. That means kidneys are damaged. They are not filtering properly from this area. GFR is damaged. Definitely in the plasma, urea level is becoming high and high. Urea level become high, because if GFR is not properly, that is filtration is not possible over here due to the damage in this area. This area is damaged and urea cannot move from this area to this area. Definitely in the plasma, urea level become high. So if it is going to decrease, then it will increase, and this will push the urea excretion. This will push the urea. So more and more urea excretion will be possible if the GFR is decreased, and this is because urea can be excreted with very less amount of water due to again recircling of this urea again and again from the inner medullary ducts to the thick ascending of the loop of Henle or the hairpin turn, and this again is going to move out, and due to the action of the ADH, which removes water from it, but the urea remains trapped inside. In the next video, or that you can say in the next lecture, I am going to discuss the role of the vasa recta. Up till now, we have covered that how the hyperosmolarity of the medulla is formed. 60% due to the accumulation or deposition of the sodium, 40% due to the urea. In the next